Welcome back everyone. So for this week I'm going to talk about our PCR product purification and restriction enzyme digestion that we're going to be doing in the lab this week. So if you remember, what we did last week was we ran our PCR and generated billions of copies of our beta-gal DNA. The problem is, in this PCR product we also have our buffer components as well as other enzymes like our Taq polymerase that we kind of want to make sure we get rid of so that we can actually use our beta-gal gene. So the way we're going to do this is by purifying our beta-gal DNA from everything else. And the way this kind of starts is actually by running everything, so our entire PCR product, through an agarose gel. So when we run our PCR product, what we'll end up seeing is here is, I guess, a really dark band that's supposed to represent our beta-gal. Maybe if our thing is contaminated, we'll get some other bands. But really, we know that this band is our beta-gal. So what we first do is we actually excise this little bit from the agarose gel and solubilize it so that we can put it into a column. So this is how our purification begins. We're going to be using a special silica membrane column. So silica is known to bind to DNA very, very well. So these silica columns, they kind of fit into little Eppendorfs. They're very small little like funnel looking tubes. And right here would be our little silica membrane. So let's pretend that's an Eppendorf. So what we try to do is we'll add our liquid into this column and it'll sit at top of on top of the column. The next thing we do is we'll actually put it into a centrifuge so that we can spin it down, and the centrifugal force will pull down the liquid, and as it pulls down the liquid, the DNA of our beta-gal gene will bind to this little silica membrane. So that's how we actually, I guess, collect our beta-gal gene. But the problem is, in this membrane might also be other things like salts that we kind of want to get rid of. And so the way we'll do this is by adding alcohol, or more specifically ethanol. So this would be the washing step of our actual purification. So we know that our beta-gal gene is in this little column, so we're going to wash all of the impurities such as proteins like Taq polymerase that might be in there, as well as any residual salts with ethanol. So what you guys really need to know is that this ethanol is kind of used to remove these residual salts by solubilizing it, but it doesn't precipitate the DNA. So the DNA still stays it leaves the DNA precipitated so that it still stays on the column. So we wash it maybe 1 or 2 times, and then we also do a dry spin because we want to make sure that we pull out all of that washing buffer, all of that ethanol out of the membrane, because ethanol will cause, I guess, further problems down the road. So it's very important that we get rid of all the ethanol. Once we were sure that we've washed it very thoroughly and removed all the ethanol, then it's time for us to elute our beta-gal gene. So we do this by applying a tris buffer back into that column. So here's that little column again. So remember, beta-gal is somewhere in here. And we're going to apply our elution buffer, which is usually tris and that contains some kind of EDTA, usually at a pH of 8. So once this buffer enters the column, it'll actually solubilize the DNA again, so that when we spin it one last time, we're able to collect it in this little Eppendorf. So that's how we're going to collect our sample. The next thing we need to do is actually digest it. So what we have is a little vector, which is our plasmid, and our beta-gal gene. What we want to be able to do is attach our beta-gal gene into the vector, kind of how you see right here. And the way we can do this is by using restriction enzymes. So restriction enzymes, they are found in bacteria. Think of them as like the bacteria's immune system. What they're used in bacteria, what they do there is they actually kind of destroy viral DNA that shows up in the bacterial, cell. So it chops up all of these DNA strands into useless fragments so that the bacteria doesn't get infected. Us in the laboratory, us trying to be clever, we saw this as a very good and useful tool, and we were able to kind of catalog different restriction enzymes and what they cut. So what you need to know is that restriction enzymes are very, very specific. They always cut at a very specific nucleotide sequence, usually around 6 nucleotides. It has to find a specific 6 nucleotides in order to cut. So like all enzymes, restriction enzymes work by shape-to-shape matching, meaning they have to, the exact sequence has to match perfectly or else the restriction enzyme won't actually cut. And these recognition sites can be anywhere from 4 to 8 nucleotides long. So these restriction enzymes are a very basic tool for biotechnology research, and they are used a lot in DNA cloning as well as DNA fingerprinting. So there are a couple ways that restriction enzymes cut. The first is a blunt cut, or to make blunt ends. So if this is our double-stranded DNA, a restriction enzyme that cuts in a blunt end will cut it straight through, giving us 2 pieces with, flat ends. Like that. And honestly, this is not really the one, it's not very useful. This is not how we want our DNA to kind of be cut. Instead, we want a little bit of an overhang, or what we like to call sticky ends. And there are actually a ton of restriction enzymes that'll cut in a very particular way, kind of like a little zigzag, so that when they cut, they leave these overhangs, like that you can kind of see right here, that we can use later on to kind of match up with our vector. So for us in, this experiment, what we're going to be using, or the 2 restriction enzymes we'll be using, are HIND3 and BAMH1. And if I recall, I believe HIN3 is going to be for our forward, and BAMH1 is going to be for our reverse. And I'll talk a little bit more about that in just a second. So remember that in your last lab report, you guys had to design primers for the beta-gal gene. So that's only one part of the actual, DNA design process. The other part is also adding a restriction enzyme. So we add restriction enzyme sequences to our primers so that we know, based on restriction enzymes that are in our plasmid. So we know that the plasmid that we're using, I believe it's PET23 or PET22A or something like that. But we know that in this vector that it has 2 restriction enzymes for, BAMH1 and HIN3. So we want to add these restriction enzymes also to our primers so that when both these get cut, they'll have the same overhang. So think of it like if this is our vector, and it's a little bit bad, but let's say these are our overhangs. So this is for, HIND. This is for BAM. And then here is our entire extended, digested beta-gal gene, also with little restriction enzyme sites for HIND3 and BAMH1. Then this site will match with that site, and this site will match with that site, so that we get a full plasmid with, our little gene somewhere stuck in the middle. So in your lab report for you guys are going to have to basically redo your restriction enzymes. So I'm going to make sure that I will check your lab reports as quickly as possible, so I can let you know if you designed your primers well. But for this lab report, we're going to basically redesign them again, and this time add those primers, basically to help you guys out to make sure that you understand how primers are actually designed. So let's say this is our beta-gal gene from start to end. So we designed a primer. So this is our forward primer. That's supposed to match up with a little segment of our gene. And what we also must add now are the restriction enzyme sites. So this right here. So that is a restriction enzyme site, or the recognition site. So this is those 6 nucleotides, these right here. So hint 3, so this is what you would see for your forward. Oops. So this is what you would see right here. Another important thing to note is that there needs to be a couple flanking bases right here. So these are usually, I think, maybe 3 to 4 just random base pairs that can just link up right here, so that when our little restriction enzyme comes, it actually has an easier time of binding. If this little, if these flanking bases aren't there, then when the restriction enzyme comes, it might bind and then kind of fall off just because it doesn't have enough room. So that's what you guys need to put in for your next lab report. Your primer that matches up with the gene, along with the recognition site for, I guess, whatever restriction enzyme. So since this is the forward, it's going to be hint 3, as well as 3 to 4 flanking bases, just so that the recognition site can bind. Notice how I'm only kind of putting it onto one side. For the reverse, we have the same thing. We have our primer. Right here, we have our recognition site and then the flanking bases. So when you write it in your report, it'll look something similar to this, just like, so if this is our forward, then this would be our reverse. So remember, it's the reverse complement. So I don't know if that was able to help anyone, but if you guys have any more questions on designing these primers, please feel free to email me. Thank you.